The world first pig kidney transplant was a huge breakthrough, but is it the future? Christopher Rouge of Conversation reports. In a world first, we heard last week the US surgeons had transplanted a kidney from a gene-edited pig into a living human. News reports said the procedure was a breakthrough in xenotransplantation, when an organ, cells, or tissues are transplanted from one species to another. One biotech CEO says gene-edited pigs promise an unlimited supply of transplantable organs. Champions of xenotransplantation regard it as a solution to organ shortages across the world. In December 2023, 1,445 people in Australia were on the waiting list for donor kidneys. In the United States, more than 89,000 are waiting for kidneys. Not everyone, though, is convinced transplanting animal organs into humans is really the answer to organ shortages, or even if it's right to use organs from or other animals in this way. There are two clinical barriers to the procedure's success: organ rejection and the transmission of animal viruses to recipients. But in the past decade, a new platform, the technique known as CRISPR-Cas9, often shorter to CRISPR, has promised to mitigate these issues. What is CRISPR? CRISPR gene editing takes advantage of a system already found in nature. CRISPR's genetic scissors evolved in bacteria and other microbes to help them fend off viruses. Their cellular machinery allows them to integrate and ultimately destroy viral DNA by cutting it. In 2012, two teams of scientists discovered how to harness this bacterial immune system. This is made up of repeating arrays of DNA and associated proteins known as Cas-CRISPR-associated proteins. When they use a particular Cas protein, Cas9, with a guide RNA made up of a singular molecule, they found they could program the CRISPR-Cas9 complex to break and repair DNA at precise locations as they desired. The system could even knock in new genes at the repair site. In 2020, the two scientists leading these teams were awarded a Nobel Prize for their work. In the case of the latest xenotransplantation, CRISPR technology was used to edit 69 genes in the donor pig to inactivate viral genes, humanize the pig with human genes, and knock out the harmful pig genes. A busy time for gene editing xenotransplantation. While CRISPR editing has brought new hope to the possibility of xenotransplantation, even recent trials show great caution is still warranted. In 2022 and 23, two patients with terminal heart diseases who were ineligible for traditional heart transplants were granted regulatory permission to receive a gene-edited pig heart. These pig hearts had 10 genome edits to make them more suitable for the transplanting into humans. However, both patients died within several weeks of the procedures. Earlier this month, we heard a team of surgeons in China transplanted a gene-edited pig liver into a clinically dead man with family consent. The liver functioned well up until the 10th day limit of the trial. How is this latest example different? The gene-edited pig kidney was transplanted into a relatively young, living, legally competent and consenting adult. The total number of genes edits made up to the donor pig is very high. The researchers report making 69 edits to inactivate viral genes, humanizing the pig with human genes, and to knock out harmful pig genes. Clearly, the race to transform these organs into viable products for transplantations is ramping up. From biotech dream to clinical reality. Only a few months ago, CRISPR gene editing made its debut in mainstream medicine. In November, drug regulators in the United Kingdom and the US approved the world's first CRISPR-based genome editing therapy for human use, a treatment for life-threatening forms of sickle cell disease. The treatment, known as Casgevy, used CRISPR-Cas9 to edit the patient's own blood, bone marrow stem cells, by disrupting the unhealthy gene that gives red blood cells their sickle shape. The aim is to produce red blood cells with a healthy spherical shape. Although the treatment uses the patient's own cells, the same underlying principle applies to recent clinical xenotransplants. Unsuitable cellular materials may be edited to make them therapeutically beneficial in the patient. We'll be talking more about gene editing. Medicine and gene technology regulators are increasingly asked to approve new experimental trials using gene editing and CRISPR. However, neither xenotransplantation nor the therapeutic applications for this technology lead to changes to the genome that can be inherited. For this to occur, CRISPR edits would need to be applied to the cells at the earliest stage of their life, such as to early stage embryonic cells in vitro in the lab. In Australia, intentionally creating heritable alterations to the human genome is a criminal offense carrying 15 years imprisonment. No jurisdiction in the world has laws that expressly permits heritable human genome editing. However, some countries lack specific regulations about the procedure. Is this the future? Even without creating inheritable gene changes, however, xenotransplantation using CRISPR is in its infancy. For all the promise of the headlines, there is not yet one example of a stable xenotransplantation in a living human lasting beyond several months. While authorization for this recent U.S. transplant has been granted under the so-called compassionate use exemption, conventional clinical trials of pig-human xenotransplantation have yet to commence. But the prospect of such trials would likely require significant improvements in current outcomes to gain regulatory approval in the US or elsewhere. By the same token, regulatory approval of any off-the-shelf xenotransplantation organ, including gene-editing kidneys, would seem some way off. And this is from the conversation here under Creative Commons by Christopher Rugg, Law Lecturer, University of Sydney, on Science Alert. Please leave your comments, and thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.